Welcome back to The Deep Dive, the show that gives you the ultimate shortcut to expertise. We take these, incredibly dense concepts, these complex chapters, and we turn them into knowledge you can actually use.
And today, we are prying open the door to the core engine room of the eukaryotic cell. We're taking an intensive look at aerobic respiration and the mighty mitochondrion.
Our mission today is to get way beyond that basic high school concept of the powerhouse of the cell.
Oh, absolutely. We're going to master the precise mechanics of how energy is converted. We'll follow the energy from ancient evolutionary struggles right down to this amazing molecular motor that literally drives life.
We'll be looking closely at every structure, every pathway, and every human connection mentioned in our sources.
Exactly. So to really get this, to appreciate the mitochondrion, we actually need to go back in time, way back.
How far back are we talking?
About 3.5 billion years, back to when Earth's atmosphere was what scientists call a reduced atmosphere.
Meaning no oxygen.
Right. It was mostly molecular hydrogen, ammonia, water vapor. And in that world, life was simple. The organisms were all anaerobes.
They didn't need oxygen.
They couldn't use it. They survived using these really basic, kind of inefficient pathways, things like glycolysis and fermentation. Critically, molecular oxygen just wasn't around.
But then something happened. Life invented a process that would change the planet forever. And for most life at the time, it was a total catastrophe.
That's the oxygen catastrophe. It started, about 2.7 billion years ago, and it was triggered by a new kind of microbe, the cyanobacteria.
And what did they do that was so revolutionary?
They perfected photosynthesis. They figured out how to use sunlight to split water molecules, and the byproduct of that process was molecular oxygen, O2.
And O2 is actually a very reactive, very dangerous molecule.
It's a poison. As the levels rose, it started attacking everything. DNA, lipids, proteins. It was toxic to almost all existing life.
So it created this immense evolutionary pressure. Adapt or die.
And most life perished. But some organisms, some lineages, found a way to not just survive the poison, but to actually use it.
Which was a huge advantage.
A monumental advantage. See, anaerobic metabolism is really inefficient. When an anaerobe breaks down food, it spits out products like ethanol or lactic acid.
Which still have a ton of energy locked inside them.
Exactly. They leave most of the fuel on the table. But the aerobes, the survivors, they evolved these new pathways that used oxygen as the final electron acceptor.
Letting them burn their fuel completely.
All the way down. They could take carbohydrates, fats, proteins, and oxidize them completely to just carbon dioxide and water. Harmless, energy-depleted waste.
An energy payoff for doing that is massive.
It's exponential. They could extract so much more energy from the same piece of food. And this whole process, this huge energy extraction machine, is housed inside the mitochondrion in eukaryotes.
And the mitochondrion itself is a direct product of that history.
It is. The theory of endosymbiosis is, the leading explanation. An ancient aerobic bacterium was engulfed by, or took up residence in, an anaerobic host.
And they struck a deal.
A mutually beneficial relationship. And the structure, the genetics, everything about the mitochondrion today is a legacy of that billion-year-old merger.
Okay, so let's fast forward to the present and look at this structure. When you see a mitochondrion in a textbook, you get this image of a little bean, maybe 1 to 4 micrometers long.
Right, that classic isolated bean-shaped organelle. But the reality, when you see it in a living cell, is often way more dynamic.
What do you mean?
Well, often mitochondria form this giant, highly branched, interconnected tubular network. It's sometimes called a reticulum.
So like a web spreading through the cell's cytoplasm.
Exactly. And it's not static. These things are constantly moving, changing shape, and undergoing two opposite processes. Fusion, which is merging together.
And fission, splitting apart.
Right. And the way they split, the fission mechanism, is a fantastic example of how different parts of the cell coordinate. It actually involves another major organelle.
The endoplasmic reticulum, the ER.
Yeah, this was a really surprising discovery. Fission doesn't start with the mitochondrion itself. It starts when these thin tubules from the ER contact and wrap around the mitochondrion.
Like a lasso or a noose?
It acts just like that, like a guiding track. Then proteins from the cytosol are recruited to that spot to finish the job. Specifically, a soluble protein called GRP1.
And GRP1 is part of the dynamine family of proteins, right? How does it actually squeeze the organelle in half?
GRP1 proteins assemble into a ring, a tight helix, around the mitochondrion, right where the ER marked it. Then, by using energy from GTP, that helix constricts. It tightens like a noose and physically pinches the organelle into two separate units.
So the overall look of the mitochondria in a cell, whether it's one long network or lots of little individual ones, is just the result of this constant balance.
The balance between fusion and fission. And that balance is incredibly important for the cell's health.
How so?
We know that mutations in the genes that code for the fusion machinery, for example, lead directly to inherited neurological diseases.
Because neurons are so energy hungry.
They absolutely are. They need that mitochondrial network to be efficient and correctly structured to survive.
Okay, beyond just the shape, let's talk about how much space they take up.
It's a huge amount of real estate.
Yeah.
In a typical liver cell, for instance, mitochondria account for something like 15 to 20% of the total volume of the cytoplasm.
And they aren't just scattered around randomly.
Not at all. Their placement is highly strategic. They are put right where the energy is needed most.
The classic example being the sperm cell.
It's the perfect example. If you look at a micrograph, you'll see the mitochondria are clustered, just wrapped tightly around the midpiece, right at the base of the flagellum, the tail.
The engine is right next to the motor it's driving.
Precisely. And you see similar things elsewhere. They're often found right next to oil droplets that contain fatty acids, ready to oxidize that fuel immediately.
But their job isn't just making ATP, they're much more than that.
Oh, they're essential metabolic hubs. They're involved in making certain amino acids and, really importantly, in synthesizing heme groups.
Heme groups. That's the iron-containing part of hemoglobin and also the cytochromes we'll talk about later.
Exactly. And maybe one of their most critical roles for cell signaling is as regulators of cytosolic calcium ions, K2 plus PAT.
How do they regulate calcium?
They act like a sponge. They can rapidly take up excess calcium from the cytosol or release it when needed, which helps buffer and control the concentration.
And since calcium is a major signal for everything from muscle contraction to releasing neurotransmitters, that's a vital role.
Absolutely. And one last thing. They play a direct role in regulating programmed cell death, or apoptosis. We'll see later that certain proteins from inside the mitochondrion can act as a kill switch for the cell.
Okay, so let's dissect the physical architecture. It has two membranes and two separate compartments.
Let's start on the outside. The outer mitochondrial membrane, it's the boundary. And it's filled with these special integral proteins called porins.
Porins, like pores.
Essentially, yes. They form these large channels shaped like a barrel of beta strands. It's actually a structure you see in the outer membranes of some bacteria.
And what does that mean for permeability?
It means the outer membrane is pretty much freely permeable to small molecules. Things like ATP, NAD, coenzyme A, they can all pass right through those channels.
So it's not much of a barrier.
To small things, no. Though there is evidence the porins can close, so there's some regulation. But in contrast, the inner mitochondrial membrane is the real gatekeeper.
This one is highly impermeable.
Remarkably so. To get into the matrix, basically every molecule and ion needs a dedicated, highly specialized transporter protein.
And its structure reflects its ancient bacterial origins.
It really does. It has a super high protein-to-lipid ratio, more than 3-to-1 by weight. It's also rich in an unusual phospholipid called cardiolipin.
Which is characteristic of bacterial membranes.
Right, and it has virtually no cholesterol, which is very different from most other eukaryotic membranes.
And even within this inner membrane, there are different regions.
Two main domains. First is the inner boundary membrane. It sits right inside the outer membrane, and it's where most of the machinery for importing proteins is located.
And then there are the cristae.
Yes, the cristae. These are the elaborate folded sheets that project deep into the interior of the organelle.
This is what gives it that classic wrinkly look on the inside. And it's all about surface area, right?
That's all about surface area. The cristae can increase the surface area of the inner membrane by up to 5 times. And that massive area is needed to house the thousands of copies of the electron transport chain and ATP synthesis machinery.
And these membranes create two distinct spaces.
Correct. First, the matrix. That's the innermost compartment. It's like a dense gel, just packed with soluble proteins, up to 500 milligrams per milliliter. This is where the TCA cycle happens.
And the second space.
The intermembrane space. It's the region between the inner and outer membranes. And as we mentioned, it holds some key proteins that, if released, can trigger that cell suicide program, apoptosis.
Okay, finally, let's touch on that evolutionary relic, the mitochondrial genetic system. What's actually left of that ancient bacterium's DNA?
Inside the matrix, you find ribosomes and several copies of a small circular DNA molecule, the mtDNA.
And in humans, this tiny genome codes for what?
Only 13 polypeptides?
Yeah.
And they are all very hydrophobic subunits of the big protein complexes in the inner membrane, the ones we're about to discuss.
So they're made right there on site because they're so hard to transport?
That seems to be the idea.
Yeah.
The mtDNA also codes for 2 ribosomal RNAs and 22 transfer RNAs, everything needed to run its own little protein synthesis operation inside the organelle.
And there are other clues to its origin, right?
Oh yes, the mitochondrial RNA polymerase, the enzyme that transcribes the DNA, is a single subunit enzyme. This is totally different from the big, complex polymerases in the cell nucleus, but it looks a lot like the ones used by bacteriophages.
It's just another piece of the puzzle pointing back to that prokaryotic ancestor.
Absolutely. And because mtDNA is small and it's inherited strictly from the mother, its sequences have become this incredibly powerful tool for tracing human migration and evolution.
All right, we have the layout of the power plant. Now let's talk about the fuel and how it gets processed. It all starts for glucose anyway, out in the cytosol with glycolysis.
Right. Glycolysis gives you a quick small burst of energy, a net of 2 ATPs. But the main products it sends forward are pyruvate and NADH. And crucially, almost all of the original energy from the glucose is still locked up in that pyruvate.
So if oxygen is available, that pyruvate needs to get into the mitochondrial matrix. How does it cross that super impermeable inner membrane?
It uses a specific transporter protein. And once it's inside the matrix, it's immediately grabbed by this enormous enzyme complex, the pyruvate dehydrogenase complex.
And what does that complex do?
It performs what's called an oxidative decarboxylation. It snips off one carbon as CO2, and it attaches the remaining 2 carbon unit, an acetyl group, to something called coenzyme A.
Forming acetyl-CoA.
Exactly. And in the process, it generates another molecule of high-energy NADH.
And acetyl-CoA. This is the universal entry ticket to the central metabolic factory.
It is. It's the key that unlocks the tricarboxylic acid cycle, or TCA cycle. You might also know it as the Krebs cycle, or the citric acid cycle.
And all the enzymes for this are just floating around in the matrix.
Almost all of them are soluble proteins in the matrix.
Yeah.
There is one really important exception, though. Succinate dehydrogenase. It's actually physically bound to the inner mitochondrial membrane.
Okay, so how does this cycle kick off?
It starts when that 2-carbon acetyl-CoA joins with a 4-carbon molecule called oxaloacetate. They condense to form a 6-carbon molecule, citrate.
And then the cycle begins, and during one turn, 2 carbons are kicked out as CO2.
Right. The 2 carbons that came in with acetyl-CoA are completely oxidized and released. And at the very end of the loop, the 4-carbon oxaloacetate is regenerated, ready to grab the next acetyl-CoA. It's a true cycle.
And what's the main output? It's not really ATP, is it?
Not directly. The main output is reduced coenzymes. During one turn of the cycle, you get 4 separate redox reactions that generate 3 molecules of NADH and 1 molecule of FADH2.
So it's loading up these electron carriers.
It's all about loading up those carriers. You do get 1 molecule of GTP, which is easily converted to ATP, but the vast majority of the energy is now stored in the electrons carried by NADH and FADH2.
Now, the TCA cycle is often called a point of metabolic convergence. What does that mean?
It means it's the central funnel for pretty much all energy-providing macromolecules. It doesn't matter if you eat carbs, proteins, or fats. They all get broken down into intermediates that feed right into the TCA cycle.
So let's take fats as an example. How do they feed in?
Fatty acid oxidation. This process also happens right there in the matrix. Fatty acids get broken down 2 carbons at a time in a process that generates 1 acetyl-CoA for each 2-carbon chunk.
So it feeds directly into the start of the cycle.
Correctly. And each round also produces 1 NADH and 1 FADH2. This is why fats are such an incredibly rich source of energy compared to carbs.
And what about proteins?
A similar story. Amino acids are broken down into various fragments that are identical to intermediates already in the TCA cycle. They might become alpha-ketoglutarate or succinyl-CoA. They just jump into the cycle at different points.
Okay, so all roads lead to the TCA cycle, but there's a problem we need to solve. That NADH that was made way back in the cytosol during glycolysis, it's stuck outside.
It is. The inner membrane is impermeable to it. So the cell uses these brilliant shuttle systems.
They don't move the NADH itself.
No, they move its high-energy electrons. They pass the electrons to a smaller molecule that can cross the membrane, which acts like a little ferry.
And there are 2 main shuttles for this.
All right, the first is the malate aspartate shuttle. This one's a bit more complex, but it's the high-yield route. It effectively transfers the electrons to a carrier that, once inside the matrix, reduces NAD+ back into NADH.
So the electrons maintain their high energy state.
Exactly. They get to enter the next stage at the highest possible energy level.
Yeah.
The other option is the glycerol phosphate shuttle.
And this one sacrifices a little energy for speed.
That's a good way to put it. Here, the cytosolic NADH passes its electrons to a substrate that moves them across the membrane. But on the inside, those electrons are immediately transferred to FAD, making FADH2.
And FADH2 is a slightly lower energy carrier.
It is. This FADH2 feeds its electrons into the chain at a later point, bypassing the first proton pumping step. So you get a little less ATP out of it, but the process is faster.
So now finally we have this huge stockpile of high-energy electrons carried by NADH and FADH2, all ready to go inside the matrix. This brings us to the grand finale.
Oxidative phosphorylation. It's a two-step process, and the whole idea was revolutionary when Peter Mitchell proposed his chemiosmotic hypothesis back in 1961.
So step one.
Step one is the electron transport chain, or ETC. The high-energy electrons from NADH and FADH2 are passed down a series of carriers, like a cascade, embedded in the inner membrane.
And the final destination for those electrons is oxygen.
O2 is the final electron acceptor, and as the electrons move down this chain, they release energy. And that energy is used to power the protein complexes to pump protons, H+ ions, from the matrix out into the inner membrane space.
So you're converting the chemical energy of the electrons into the potential energy of a concentration gradient.
Electrochemical gradient, precisely. And that leads to step two, chemiosmosis. You've built up this high concentration of protons in the inner membrane space, and they desperately want to flow back into the matrix.
But they can't just cross the membrane.
No, they have to pass through a specific channel, a specialized enzyme called ATP syntase. And the controlled flow of those protons back down their gradient is what powers the synthesis of ATP.
And the final yield is incredible.
It is. For every NADH, you get enough energy to make about 3 ATPs. For FADH2, which enters a step later, you get about 2 ATPs.
So when you add it all up for 1 molecule of glucose,
You're looking at a total yield of roughly 36 ATPs. Compare that to the 2 net ATPs you get from glycolysis alone. It's a huge difference.
No, let's connect this huge energy difference to something practical, like human exercise. The source material has a great section on this.
Right. Muscle contraction is incredibly energy intensive, but our actual stores of ATP in the muscle are tiny, enough for maybe a 2 to 5 second burst of all-out effort.
So for anything longer, you need to regenerate it, and fast. The first backup system is creatine phosphate.
The phosphagen system. Creatine phosphate, or CRP, can rapidly donate its phosphate group to ADP, making more ATP. But that only buys you about another 15 seconds.
So for sustained activity, you have to start oxidizing fuel, and the type of fuel depends on the muscle fiber type.
Exactly. You have your fast twitch fibers. These are built for power sprinting, weightlifting. They don't have a lot of mitochondria, so they rely mainly on anaerobic metabolism, on glycolysis.
Which is less efficient per glucose, but it produces ATP very, very quickly.
Right. It's great for those short, intense bursts, but there's a downside.
Pig.
And fast. You burn through your glycogen stores, and you produce a lot of lactic acid. Now, that lactic acid is necessary to regenerate the NAD+ to keep glycolysis going.
But it also makes the muscle acidic.
Very acidic. The pH can drop from a resting 7.0 down to 6.35. And that acidity plus the fuel depletion is what causes that muscle fatigue and cramping.
On the other hand, you have the slow twitch fibers, the marathon runner's fibers.
These are packed with mitochondria. They're designed for sustained aerobic activity like jogging. They generate less force, but they can produce ATP continuously, efficiently, and without the lactic acid buildup.
And they're smart about their fuel use.
They're incredibly economical. They start out using glucose and glycogen. But after just 20 minutes of vigorous aerobic exercise, they start to shift. Up to 50% of the calories being burned can come from free fatty acids released from fat tissue.
Sparing the limited carbohydrate stores for later.
Exactly. And then you have the ultimate aerobic machine, the heart.
It has to be aerobic. It can't function anaerobically for more than a few seconds.
It's completely dependent on it. And to support that, an astonishing 40% of the entire cytoplasmic space in a human heart muscle cell is just mitochondria. It really shows you how vital this organelle is.
All right, let's zoom in now. Let's get into the physics of this electron transfer. To really get it, we need to understand oxidation reduction potentials.
Or E0 prime. The core concept is electron transfer potential. So reducing agents are electron donors. A strong reducing agent, like NADH, holds its electrons loosely. It has a high potential to give them away.
And oxidizing agents are electron acceptors.
Right. A strong oxidizing agent, like oxygen, holds the electrons very, very tightly.
And we measure this potential in volts relative to a standard, which is the hydrogen couple, set at 0.
Correct. And by convention, the strong electron donors, the ones that want to give electrons away like NADH, get a very negative potential, around minus 0.32 volts,
while the strong acceptors, like oxygen, get a highly positive potential.
plus 0.82 volts.
Hmm.
So the electrons are flowing from a very negative potential to a very positive one, a huge drop.
And that drop in potential lets us calculate the free energy release, how much energy is available from that transfer.
And it's a massive number. For a pair of electrons moving all the way from NADH to oxygen, the free energy change is minus 52.6 kilocalories per mole.
That is a lot of energy.
It's a huge amount of energy, far more than you need to make one ATP. So the cell's challenge is to not release that all at once like an explosion, but to capture it in small, manageable steps.
And that's the job of the carriers in the electron transport chain. There are 5 main types.
Right. First, you have the flavoproteins. These use FAD or FMN as their prosthetic group. They're versatile. They can accept and donate 2 electrons and 2 protons.
Complex I and complex II are examples.
Exactly. Second, you have the cytochromes.
These use heme groups, with an iron atom in the middle.
And that iron atom can flip between the F2 plus and F3 plus states, so they transfer 1 electron at a time.
Third, we have copper atoms.
Found in the final complex, complex 4. They also transfer a single electron, flipping between C1 plus and C2 plus there.
Fourth, the mobile carrier ubiquinone.
Also called Q. This one's different. It's a small, lipid-soluble molecule, not a protein. It can diffuse rapidly within the membrane, and it carries 2 electrons and 2 protons.
And finally, the iron-sulfur proteins.
These have iron atoms linked to inorganic sulfide and cysteine residues. They form these little clusters, but the whole unit typically only transfers 1 electron at a time.
So the whole power of the ETC comes from arranging these carriers in a very specific sequence.
A sequence of increasing redox potential. The electrons flow downhill from components with a highly negative potential to those with a highly positive one. A constant, regulated energy drop.
And how did scientists figure out that exact sequence?
Using inhibitors. Respiratory poisons. For example, if you add rotenone, it blocks the chain at complex 1. You can then see that everything before the block gets stuck with electrons, it becomes reduced, and everything after it stays oxidized.
So by using different poisons that block at different points, they could map the whole pathway.
That's exactly how they did it. And when you isolate these components, you find they organize into 4 huge membrane-spanning structures, complexes 1, T, 3, and 4.
With ubiquinone and cytochrome C acting as mobile shuttles between them.
Right. And 3 of these complexes, A, 3, and 4V, are the actual proton pumps. These are the coupling sites where the energy released from electron transfer is harnessed to move protons from the matrix to the inner membrane space.
Okay, let's walk through them. Complex I, NADH dehydrogenase.
This thing is a behemoth. It's L-shaped, has 45 different subunits in mammals. It takes 2 electrons from NADH and passes them to ubiquinone, or UQ. And in that process, it pumps 4 protons across the membrane.
And the mechanism for that pumping is incredible. It's like a machine.
The proposed model is called the steam engine mechanism. The idea is that the energy from the electron transfer causes a specific helix in the membrane-spanning part of the complex to physically shift its position.
Yeah, it's like a piston.
Like a molecular piston. And that movement drives conformational changes that force protons through these half channels and out the other side. It's literally converting electrical energy into mechanical movement to pump ions.
And we have to say again, when this complex fails, the consequences are severe.
They are. We know specific toxins that inhibit complex I can cause Parkinson's-like symptoms. And patients with Parkinson's disease show a selective decrease in complex activity. It's a major point of vulnerability.
Okay, next is complex 2, succinate dehydrogenase.
This is the one that's also part of the TCA cycle. It feeds the lower energy electrons from FADH2 into the chain, passing them to ubiquinone.
And the key thing to remember about complex 2 is
it does not pump protons. That's why FADH2 yields less ATP than NADH. It bypasses that first pumping site.
Then we have complex 3, cytochrome BC1.
Complex 3 takes the electrons from ubiquinol and passes them to the next mobile carrier, cytochrome C, and it pumps 4 protons across the membrane using a clever mechanism called the Q cycle.
And finally, we get to the end of the line. Complex 4, cytochrome C oxidase. This is where oxygen comes in.
And this is the most dangerous, most sensitive step. Complex 4 has to safely manage the transfer of 4 electrons, one at a time, to a single O2 molecule to turn it into 2 molecules of water.
If it messes up and releases a partially reduced oxygen molecule, that's a reactive oxygen species, a dangerous radical.
which can cause immense cellular damage. So to prevent that, it has this very special active site, a heme A3 cub binuclear center. It holds onto the oxygen molecule tightly until all 4 electrons have been delivered, ensuring a clean, complete reduction.
And complex 4 also contributes to the proton gradient in 2 ways.
That's right. For every O2 it reduces, it takes a total of 8 protons from the matrix. 4 of them are substrate protons that get used to actually make the 2 water molecules.
And the other 4?
The other 4 are pumped protons. They're actively translocated across the membrane. So both sets of protons contribute to the total gradient.
Okay, so this whole process has been pumping protons, building up this powerful gradient. Let's quantify it. The proton motive force.
Or delta P.
Okay.
And because you're moving charged particles, positive protons, across a membrane, it's an electrogenic process. It has 2 components.
First is the voltage.
The voltage, or delta Ci, that's the charge separation. The intermembrane space becomes positive, the matrix becomes negative, and in mammals, this voltage component is the dominant part, maybe 80% of the total force.
And the second component is the pH gradient.
The delta pH, that's the chemical concentration difference. The intermembrane space gets more acidic, a lower pH than the matrix. That accounts for the other 20%. All told, the total force is about 220 millivolts. A lot of stored energy.
And that stored energy can be destroyed by molecules called uncoupling agents.
The classic example is a drug called dinitrophenol, or DNP. What DNP does is it makes the intermembrane leaky to protons. It acts like a shuttle, grabbing protons in the intermembrane space and carrying them right back into the matrix.
So it bypasses the ATP synthase completely?
It short-circuits the system. Electron transport keeps running like crazy, but you stop making ATP.
And all that energy from the electron transport, instead of being stored in ATP, it just gets released as
Heat. Pure heat. This is why DNP was briefly and tragically used as a diet pill. People's bodies would burn fat and glucose, furiously trying to make ATP, but all the energy was wasted as heat, leading to fatal hyperthermia.
But the body can do this on purpose with its own uncoupling proteins.
Yes, UCP1 is the famous one. It's found in brown adipose tissue, or brown fat. And its job is non-shivering heat production. It intentionally lets protons leak back into the matrix to generate heat and keep you warm.
And the other UCPs are thought to act more like a safety valve.
A protective function, exactly.
Oh.
If the proton motive force gets too high, it can actually stall the ETC, which leads to electron leakage and the production of those dangerous reactive oxygen species. The UCPs can bleed off a little of the pressure to prevent that.
All right, we have built the gradient. Now let's look at the machine that uses it, the ATP synthase.
This is one of the most stunning pieces of biological engineering in existence. Early electron micrographs showed these little mushroom-shaped things sticking off the inner membrane.
And researchers found that the head part, the F1 unit, could actually break down ATP, an ATPase.
Which seemed backwards for something that's supposed to make ATP, but it just confirmed that enzymes are reversible. Under normal conditions, with a proton gradient, it makes ATP. Without the gradient, it runs in reverse and breaks it down.
So let's break down this amazing structure.
It has 2 main parts. The F1 head, which sticks into the matrix and has the catalytic sites, and the S0 base, which is embedded in the membrane and contains the proton channel.
The F1 head has a very specific arrangement of subunits.
It's made of 3 alpha and 3 beta subunits, arranged like segments of an orange. The 3 catalytic sites for making ATP are on the beta subunits, and running right through the middle of them is the central stalk, made mostly of the gamma subunit.
And the F0 base is where the protons flow through.
Right. It's made of a ring of C subunits, which acts as the rotor, and then other subunits that form the channel and stator.
Now, the mechanism for how this works is Paul Boyer's binding change mechanism. This was a radical idea.
It completely changed the game. The key insight is this. The energy from the proton flow is not used to actually form the chemical bond of ATP.
Wait, really? What is it used for then?
It's used to release the ATP after it's been made. It turns out that when ADP and phosphate are bound tightly to the enzyme's active site, they spontaneously form ATP with no energy input needed. The hard part isn't making it. It's getting the product to leave.
That seems so counterintuitive.
It does, but it's about equilibrium. The energy from the protons is used to force a change in the shape, the conformation, of the active site. This change in shape lowers the site's affinity for ATP, essentially kicking the product out so the next cycle can begin.
So Boyer proposed that the three catalytic sites cycle through three different shapes.
Exactly. The L, or loose state, where ADP and phosphate combined, the T, or tight state, where they're bound so tightly that they form ATP, and the O, or open state, which has such a low affinity that the newly made ATP is released.
And what drives the sites to cycle through L, T, and O?
This is the most amazing part, rotational catalysis. The central gamma subunit, the stalk, physically rotates, and as it turns, its asymmetric shape pushes against each of the three beta subunits in turn, forcing them to change shape from L to T to O.
So it's a motor. You're converting the electrical energy of the proton gradient into the mechanical energy of rotation.
And then into the chemical energy of the released ATP bond. It's an energy transduction machine.
And this incredible idea was proven by some brilliant experiments. First, the structural work.
John Walker's X-ray crystallography showed that the gamma subunit was indeed highly asymmetric, and it was positioned perfectly to interact differently with the three beta subunits, just as the model predicted.
But the real smoking gun was seeing it move.
Masasuke Yoshida's lab did that. They stuck the F1 part to a slide and attached a long fluorescent actin filament to the top of the gamma subunit. Then they fed it ATP to make it run in reverse.
And they watched the actin filament spin like a propeller.
In discrete 120-degree steps. It was direct, visual proof of rotation. Later, they even forced it to make ATP by using magnets to spin the gamma subunit. A full 360-degree turn made exactly 3 ATPs.
So how does the F0 base turn that proton flow into rotation?
It's the ring of C subunits, the rotor and the stationary subunit. A subunit has two half channels for protons, one that opens to the inner membrane space and one that opens to the matrix. They don't connect.
Okay, so how does that make the ring turn?
A proton enters from the inner membrane space through the first half channel. It binds to an acidic residue on one of the C subunits in the ring. This neutralizes the charge and causes the whole ring to rotate slightly.
Carrying that proton with it.
Exactly. It carries it almost a full circle until that C subunit lines up with the second half channel, the one leading to the matrix. The proton is then released into the low concentration matrix, the residue becomes charged again, and the cycle continues.
A constant flow of protons drives a constant rotation. It's incredible.
It really is. And that proton motive force isn't just for making ATP, it powers other things too.
Like what?
Like transporting the new ATP out into the cytosol and bringing fresh ADP in. That's done by the adenine nucleotide translocase. It also drives the import of phosphate and a lot of the proteins the mitochondrion needs.
Okay. Our source material also points us to another organelle involved in oxidative metabolism, the peroxisome.
Right. Peroxisomes are much simpler. They're basically small, membrane-bound vesicles filled with over 50 different enzymes.
And they share a few features with mitochondria.
They do. They also form by splitting from existing ones, and they have to import all their proteins from the cytosol.
Their name comes from their most famous job, handling hydrogen peroxide.
H2O2. It's a toxic byproduct of certain oxidative reactions that happen inside them. To deal with it, peroxisomes are packed with an enzyme called catalase, which very rapidly breaks the hydrogen peroxide down into harmless water and oxygen.
But they also have key roles in lipid metabolism.
Very important roles. They are the primary site for breaking down very long-chain fatty acids. These extra long fats have to be shortened in the peroxisome before they can be sent to the mitochondrion for the final burn.
And they're involved in synthesis, too.
Yes, they're essential for making a special class of phospholipids called plasmalogens, which are critical for the myelin sheaths that insulate our nerve cells.
Okay, so this brings us to the final part of our deep dive, the human perspective. What happens when these systems go wrong? Let's start with mitochondrial diseases.
Because mitochondria are so crucial for high-energy tissues, these diseases primarily affect the brain and muscles. You see symptoms like profound muscle weakness, fatigue, and all sorts of neurological problems.
And the genetics are unique.
They are maternally inherited. You get all of your mitochondria from your mother's egg cell. This often leads to a situation called heteroplasmy, where a person has a mix of healthy and mutated mtDNA in their cells.
And symptoms only appear when the number of bad mitochondria in a tissue crosses a certain threshold.
Exactly. And that mtDNA is very vulnerable to mutation. It sits right next to the electron transport chain, which inevitably leaks some of those highly reactive oxygen species.
So the mutation rate is much higher than in our nuclear DNA.
More than 10 times higher. And this led to the idea that the gradual accumulation of these mtDNA mutations could be a driver of aging and neurodegenerative disease.
And there's a strong link to Parkinson's disease.
A very strong link. It was first discovered, tragically, in drug users who developed sudden, severe Parkinson's. The contaminant in their drugs was a chemical called MPTP.
Which turned out to be a potent inhibitor of complex I of the ETC.
Exactly. And further research confirmed it. Parkinson's patients show a selective decrease in complex I activity in the affected part of the brain. Environmental toxins that also inhibit complex I, like the pesticide rotenone, are also implicated as risk factors.
What about the broader link to aging? There was a key experiment with mutator mice.
Right. These mice were engineered to have a faulty mtDNA replication enzyme, so their mtDNA accumulated mutations at a very high rate.
And what happened to them?
They aged prematurely, they had a shortened lifespan, graying hair, osteoporosis, all the classic signs of aging. This showed that accumulating mtDNA damage is sufficient to cause premature aging.
But the story is a bit more nuanced.
It is. While it's sufficient, the sources suggest it may not be required for normal aging. The relationship is complex, and it's still an active area of research.
Okay, let's switch to diseases of the peroxisome, Zellweger syndrome.
This is a devastating, often fatal disease. The core problem is that the peroxisomal enzymes are made correctly in the cytosol, but they fail to get imported into the peroxisome.
So you're left with these empty membrane ghosts.
Exactly. The enzymes are there, but they're in the wrong place, so they can't function.
And the other major one is X-linked adrenoleukodystrophy, or XALD.
XALD is a progressive neurological disease caused by a defect in a single transporter protein in the peroxisome membrane.
The ABCD1 protein.
Right. Its job is to move those very long-chain fatty acids into the peroxisome so they can be broken down. When it's defective, these fatty acids build up in the brain and destroy the myelin sheaths of nerve cells.
But treatment for XALD has become a real success story.
It has. Early treatments had mixed results, but bone marrow transplants were successful. And more recently, gene therapy has shown incredible promise.
How does that work?
They harvest stem cells from the patient, use a virus to insert a correct, functional copy of the ABCD1 gene, and then infuse those repaired stem cells back into the patient. It's been shown to halt the progression of the disease, a really profound example of molecular medicine at work.
So we have completed this massive journey. Let's try to synthesize the key takeaways.
I think there are three big ones. First, the evolutionary context. The mitochondrion is this elegant solution to the oxygen problem, allowing for the total oxidation of fuel through that central hub, the TCA cycle.
Second, the two-step mechanism of oxidative phosphorylation. You use electron flow to build an electrochemical gradient, mostly voltage.
And then third, you use that gradient to power a physical rotary motor, the ATP synthase, which mechanically forces the release of the final ATP product. It's a machine.
And we saw this critical partnership with the peroxisome and how failures in these tiny components can lead to devastating human diseases, affecting everything from our endurance to our neurological health.
The thing that should really stick with you is just the mechanical brilliance of it all. The cell's ability to convert chemical energy to electrical potential, then to mechanical motion, and then back to chemical bonds it's just astonishing.
Thank you for joining us for this intensive deep dive into aerobic respiration and the mighty mitochondrion. We really hope this gives you clarity and genuine expertise on the topic.
Until next time, keep exploring the intricate world of the cell.